The electrical activity of the heart, which governs every heartbeat, is a finely tuned symphony of ions moving across cell membranes. These movements occur through specialized protein pores called ion channels. Understanding these channels is essential for grasping how the cardiac action potential works and how various antiarrhythmic drugs exert their effects. Sodium channels are responsible for the rapid upstroke of the action potential in non-pacemaker cells, that is, atrial and ventricular myocytes. Primary sodium channel is NaV1.5, encoded by SCN5A gene. They open rapidly in response to depolarization, allowing a massive influx of sodium ions. This causes phase 0 of the action potential. Brugada syndrome is often caused by mutations in SCN5A channel. Class 1 antiarrhythmic drugs like lidocaine or flecainide work by blocking these channels. Calcium channels bridge the gap between electrical excitation and mechanical contraction, that is, excitation-contraction coupling. L-type calcium channels. These are long-lasting channels. They open during phase 2, that is the plateau phase, allowing calcium ions to enter the cell, which triggers the release of even more calcium from the sarcoplasmic reticulum to make the heart muscle contract. T-type calcium channels. Transient channels that play a larger role in the pacemaker activity of the SA and AV nodes. Calcium channel blockers like verapamil or diltiazem are used to control heart rate and treat hypertension. Potassium channels are the reset buttons of the heart. They allow potassium ions to leave the cell, making the interior more negative again, that is, repolarization. Rapid and slow delayed rectifier channels, IKR and IKS. These are responsible for phase 3 repolarization, bringing the cell back to its resting state. Long QT syndrome. Mutations in these channels like KCNH2 for IKR or KCNQ1 for IKS delay repolarization, lengthening the QT interval and increasing the risk of toss at the point. Class 3 antiarrhythmics like amiodarone and sotalol primarily target these channels. Inward rectifier channels IK1. These maintain the stable resting membrane potential during phase 4. Funny channel related to IF current is primarily found in the pacemaker cells of the SA node. This channel is unique because it is activated by hyperpolarization, that is, when the cell voltage gets low rather than depolarization. It allows a mixed current of sodium and potassium to leak into the cell, causing a slow upward drift in voltage during phase 4. This is what allows the heart to beat automatically without an external trigger. The drug ivabradine selectively blocks this channel to slow the heart rate without affecting contractility. Here is a summary of ion movements. Phase 0 of the action potential, that is depolarization. Primary ion movement is sodium influx, and that is mediated by fast sodium channels. Phase 1, early part of repolarization, potassium efflux, that is the transient outward current ITO. Phase 2, plateau phase, there is calcium influx and potassium efflux, mediated by L-type calcium channels. Phase 3, repolarization, potassium efflux, delayed rectifier potassium current. Phase 4, resting and pacemaker potentials, sodium potassium leak that is the IF current mediated by funny channels and IK1.